Yeah, I see it.
So namaste and welcome everyone to this live webinar on COVID-19. What is the latest? We have with us our Executive Director Kavita Tiwari ji. Namaste Kavita ji. Dr. Pranav Loyala, cardiologist, and Dr. Swaminathan Iyer. Namaste Dr. Iyer ji. Thank you and welcome to this. So Sewa International has a Facebook page, Sewa International USA. So please do like and subscribe to it. And we also have a YouTube channel. So please also do subscribe to the YouTube channel to get the latest updates. So Kavita ji, would you like to introduce Dr. Loyalka first? And then from there, we can introduce Dr. Iyer. Kavita ji, please.
Okay, thank you so much, Achaleshi. Can you hear me well?
Yes, we can.
Okay, wonderful. So thank you again, Pranav, once again for coming over and helping us today. As you all probably know him very well now, Dr. Loyalka is an intervention cardiologist with the HCA network. He is the director for the Gulf Coast region. And he has been very kindly helping us moderate the many medical and technical discussions we've had to make it more simple and understandable for all of us. So Pranav, I'll go ahead and hand it over to you. Why don't you introduce Dr. Iyer, and then we can start with that. Thank you.
Thank you, Achaleshi. Thank you, Kavita ji, and welcome everyone. I think we have an interesting topic today. I'll be acting as moderator. It's on the emerging treatments for COVID-19. I have the distinct pleasure of having Dr. Swaminathan Iyer. He's a professor of the Department of Cancer Medicine at MD Anderson, more lymphoma and myeloma. And today, I think he's very well versed in talking about the emerging treatments for COVID-19. And so I will let Dr. Iyer take it from there.
Thank you. Thank you for this opportunity, Pranav ji, for this generous introduction, and Kavita ji for making this happen within a short time, and Achaleshi ji for this, for trying to get us all together on this platform. It's a great opportunity to be here. And the disclaimer: I'm a hematologist and a transplanter, but the background is immunology. So a lot of what we will talk about sort of involves infectious diseases. But at the same time, it covers everything that we do. Dr. Loyalka is a cardiologist, a hematologist, but then we see patients today because it affects everybody. And with that, I'll give the mic back to you.
Right. So, Dr. we're talking fusion on the treatment for COVID-19 and what's possible, what's not. What might be nice is, could you give everyone just a little bit of an overview, maybe, of how the infection works? What are we exactly targeting? Why are we targeting? What therapies? There are probably over 100 medicines I read at the last count that people are trying to use. They're using serum. They're using many things. Really, I think what would be nice today is to go through what are the basic underpinnings of what we're doing, why we're doing it, what are the things that you might think hold a lot of hope, and others that perhaps won't be as strong a candidates as others.
No, I think we're learning a lot, and we're learning pretty fast. If you think about where it all started in January, I'm going to try and show you something on the screen. I hope you can see all of you can see this. And all of this started sometime in December, when in China, in Wuhan, in West China, when they started seeing cases of pneumonia, which seemed very different from the normal usual patterns. And then January 3rd, they found many more patients, and there was a cluster of infections that happened, and they isolated very quickly and found that this is very similar to the previous infection that happened in the year 2003. 2003 was the year of SARS, the severe acute respiratory syndrome virus. And this was very similar, but not the same. And then they called it SARS-CoV-2, come back to the abbreviation in a second. And clearly, the people in Wuhan realized that it's not an ordinary virus. It is causing a lot of people. Suddenly, you're seeing people in their, not just in the 60s and 70s going to the hospital, but even the people in their 40s and 50s getting admitted, and suddenly their whole hospital system was swamped. Middle January, the health authorities actually took notice, and from being a medical concern, it became a public health concern. And that's when the whole public health mitigation and containment efforts started in China. And as you can see in this picture here, and the reason why it happened is that there's no picture of the virus, but if you imagine this virus, here it is, right in the center. This virus is a virus, belongs to the family called coronavirus. Now, coronavirus is not that uncommon of a virus. It causes human infections. In MD Anderson, we see a lot of patients. I treat lymphomas and myelomas, so we do see patients who get coronavirus, and there are different strains of coronaviruses, and generally they cause very mild illness. And that's what, when people refer to as the common flu, people get some infection, get over it, there's no treatment for it, and within a week or two, people get better. But what is different about this coronavirus is that it has a very rapid penetration, not just into the nose and the sinus area, but also into the lungs. And if you can see in this picture, a healthy person will have these air going through the nose into the lungs, and the lungs have these air sacs called the alveolus, and that's where the oxygen exchange takes place. Oxygen from outside gets exchanged through the blood vessels, and that eventually goes into the left side of the heart and gets pumped out of the rest of the body, right? So that's the healthy part. But when this virus infects these air sacs very rapidly, goes from the mucosa and back into the lungs, it can latch on to the cells, get into the cell, hijack the cell, and start producing its own components. And interestingly, this is a virus, and the virus doesn't have a life of its own, right? Outside of the human body, it doesn't have a life. But once it comes across a living organism, it's able to recreate itself and propagate itself. And it has its co-conspirators, the capsule, the famous capsule which has spikes projection, which is why it's called the coronavirus. And inside this capsule is this virus which has the nucleic acid. The nucleic acid is the fundamental basis for life. It's called the DNA or the RNA. The DNA is what gives you the message, which gets translated into RNA in the cell. So this virus belongs to the category of RNA, a ribonucleic acid. And the advantage of the ribonucleic acid over DNA is that you don't have that step of going from the DNA to the RNA. It's straight away, it's able to reproduce itself. So it goes quickly into the reproducing complex called the ribosome and produce its own copies, and also the capsule, and then come out of the cell. And it does so very rapidly. And as you can see, in mild infection, it seems to be okay. But because it spread so quickly, all the alveolar sacs get infected at the same time. Some of these sacs, the cells, they die very quickly. And what happens is that by the time the immune system realizes what's going on, you have a very severe pneumonia setting up in the lungs. And that's the situation with this SARS-CoV-2 virus. As I said, January 15th or so, we realized that this is a pandemic in the making, and by mid-March, the entire world got wind of it. Iran, South Korea, Singapore, and etc. The rest is, you all know. I won't get to the public health part of it, but it's important to know that this virus is an RNA virus, and it escapes the recognition by the immune system for some part. And when it does so, the immune system tries to overcompensate. So part of the problem that happens here is not just the virus replicating, but also the immune system trying to get rid of it. In doing so, the body gets affected, the lungs get affected.
So I think that's, a good overview. So, if you're looking at this virus, why doesn't the flu virus do this? What is it that makes this virus different than the flu? If you had to look at it, why is this particular virus different?
A good question, and there are some fundamental virus-related issues and also the infectivity rate issues that are different. So the flu, the way the flu works is that it is also an RNA virus. It's a negative stranded RNA virus, whereas the coronavirus is a positive stranded RNA viruses. The difference is that the flu requires an additional step before it can copy itself. The coronavirus can straight away go to the ribosome and then make copies itself. Like you put something in the 3D printer, press the button, and out it comes, right? And that's how it happens. And that's number one. Number two is that its infectivity rate is very high, much higher than the flu. And there is something called the R naught. What is the R naught? Is the rate, the number of people that it can infect at a given point in time. There are different values and the derived values. The flu virus has a value of anywhere between 1 and 1.3. The coronavirus has a value of between 2 and 3. And I think I wanted to show you one other slide on where we are with this. And if you look at the other viruses, and this is a good snapshot of how contagious coronavirus is. It's not the most infective contagious virus. And if you look at the seasonal flu, this is the y-axis here and the x-axis on this side. And you have all these different viruses laid out for you. And at this bottom, near 0, is your seasonal flu. And the coronavirus sits right here. Ebola is very infective, it's right on the top. But it doesn't infect a lot of people. And then you have diseases like, just to give you a perspective, measles is very infective. It doesn't kill a lot of people, but it can pass on from people to people. Smallpox sits right in the center. Luckily, we don't have smallpox. Polio, both have been eradicated for them. And smallpox is eradicated. Polio, for the most part, is, except for maybe small pockets. So as you can see, coronavirus sits here. The transmission rate, you can see, are between 1.5 and 3.5. And so that's what makes it deadly. And because it overwhelms the immune system so quickly, it affects a lot of people, particularly the older people. And we come up with another number called the case fatality rate, which means the number of people who get infected and the number of people who die from it after getting infected. So the case fatality rate on an average is anywhere from 0.7 to 3.5, but it's much higher in the older people. In the older people, it's close to 15%. Interestingly, even the healthcare workers, as we're seeing in Italy, in China, is also close to 15%. So we can discuss about that later, but you can see why this is very different, because it's very efficient and it can spread from people to people very quickly.
And so, how, we know, and we've discussed on many of these forums, the prevention of coronavirus, it's almost, I had joked on a previous webinar that a lot of the things that we used to do in the old days, the namaste, the washing before you walked into the house, leaving your shoes outside, we practiced, I think, and our ancestors practiced in the old days, probably due to infections, some social distancing, and the only way to really combat these diseases. But in today's society, we're certainly not used to this. And, as we mentioned, there are a lot of potential targets. What, in your mind, has the greatest scope outside of vaccine? And certainly we can talk about vaccines and how they work, but what is on the horizon? What has the potential to work, and how would it work?
It's a great question. So I think there are many drugs over there, and but if you look at how this disease, not everybody is, the number of people who actually get to go to the hospital are about 10-15%, which means for about majority of the people, about 80% of the people, it is an illness. People may have mild symptoms or may not have any symptoms also. We're learning more about that. And about 15% of them do have symptoms. So if you take a population of 300 million, it comes down to about 10% of the population. And you're looking at, but it's a large number in the US, and you're looking at trying to mitigate and trying to curtail. So your treatment on where you, of course, is for the people sick who are in the hospital, who are in the ICU. And what are the treatments out there? So when China started doing this, they threw everything at the patient. Now, we do have some experience from the previous SARS, from another coronavirus course, which is the Middle Eastern Respiratory Syndrome virus. Now, if to understand this, you have to, a little bit of immunology. You need to understand. I'll try to make it as simple as possible. Now, the virus entering any virus for that matter, whether it is coronavirus or flu, it has to be recognized by something. And I'll give you an analogy. If you go into a country and you have to go through when you land in the airport, you go to the immigration office first and your visa gets checked and you are, right? If your papers are not right, anything that comes into the body, whether it's a virus or a bacteria, has to get recognized by this immigration officer. And it's inbuilt. And this is called the innate immunity. The innate immune system's job is to recognize these things. And interestingly, innate immune system is something we all grow up with. Even a little kid, a newborn baby has an innate immune system, because these viruses are so small that the cells are have the memory to look at these patterns of these viruses. It's called pattern receptor molecules. But this innate immunity, as soon as it recognizes the virus, it says, no, you don't belong here. I'm going to put you aside, and I'm going to bring the police in, I'm going to bring the military in. And so that's your adaptive immunity, the second arm of the immunity that produces the antibodies, that produces the CD8 cells to attack this virus. And that's normally the process. But what is this virus doing? Well, the virus sometimes gets recognized with the immigration officer, the innate immunity, but many times it escapes and it runs off and it starts reproducing in other areas, including the lungs, as I mentioned earlier. The immune system, the police go chasing after it and then start shooting the virus. They start trying to get rid of it. And when there's too much of battle that happens, and what happens is that you have a battleground, and the lung becomes the battleground. So the reason you need to understand analogy is initially when the virus comes in, your job is to contain the virus and get rid of the virus. But as the immune system evolves rapidly and exaggerated immune system response, you have to try and stop that excess force. You should ask this military and the police officers, stop, back off, and say, let's get the virus, which means your treatments are going to consist of trying to some extent suppress the immune system, and also have treatments that can the virus. So there is this drug called remdesivir. Remdesivir is a drug that very likely is going to get approved because it is a drug that works on the enzyme that makes this virus copy itself. When it goes into the ribosome and copies itself, it requires an enzyme. Remdesivir sits on it and prevents that enzyme from working. And remdesivir was in clinical trials. Interestingly, cats get a very similar disease. It's called feline infective peritonitis. Remdesivir was tested in the cats and it was found to be effective. It was also tested for Ebola. And now, luckily, we had that in the armamentarium, and Gilead had this drug. And as soon as this hit the shores of the United States, the Gilead started doing a compassionate study and it started seeing responses with remdesivir. So I think it's one of the most specific drugs for the virus. Now, there are other medications too. Of course, the most popular one is the chloroquine or hydroxychloroquine. And it's interesting that you sent me a tidbit on the history of the hydroxychloroquine and the cinchona plant, which goes all the way back to the 1600s, and how the British officers in India actually tried to make the bitter taste better by putting it in gin. And so the cinchona became the gin and tonic drink, right? So that is an interesting tidbit. But the chloroquine subsequently has been used for malaria. It works, and the chloroquine and hydroxychloroquine works in many different ways, but one of the basic ways that chloroquine works, at least in malaria, is that every cell inside the cell is like a house, right? So and there are different organelles. You have the nucleus and different parts in the cytoplasm, and everything floats in the cytoplasm. They go through a boat. It's like if you go to the theme park in Disney, you go and sit, and they go to the Pirates of Caribbean, you sit in a boat and the boat goes on a rail. Similarly, everything in the cell, whether it's an organelle or any other component, they go on a railroad inside the cell. What the chloroquine does is that it slows down those railroads. it slows down those rafts or boats within the cell. And it's a weak base, and it increases the pH of those boats, and so it stops them from moving. So think about the virus. The virus is producing itself, and then it makes copies of itself, and then it wraps itself in the bubble of the corona. And then as it's coming out, it has to come out to the same raft. What does the hydroxychloroquine and the chloroquine do? It slows them down. So chloroquine and hydroxychloroquine works by slowing them down. Doesn't have direct antiviral effects, but it seems to be effective. And especially when the viral load is low, it seems to work much better. So there are many studies that are ongoing with chloroquine. What became famous with the tweet was with the azithromycin, which is the drug that we normally give for upper respiratory tract infection. And this study was done in France. The only problem with that is that most studies have to have a placebo or a control arm, and sometimes, to know that your experimental treatment is better than the standard of care. And this one did not have two arms in the study. And there were a lot of questions that were raised. Nevertheless, I think it's still a very promising candidate. Now there are larger scale studies that are ongoing, trying to look at single agent hydroxychloroquine and also the combination. So you have remdesivir, you have the chloroquine and the hydroxychloroquine as candidates. And for antiviral therapies, there are other antiviral medications, like ribavirin was used in the past. The fasbufavir has been used for this. And so there are many antiviral medications, and even the flu medication, the oseltamivir or Tamiflu, has also been tried in this situation, but doesn't work really. The medications for HIV have been tested, and very quickly the Chinese put out a study with one of the antiretroviral HIV medication, and it didn't seem to be better at all. So these are the antiviral medications. So if you go to the immune system, we're trying to control the immune system, right? So and there are many ways to do it. The commonest way we all know as hematologists is to give steroids. But the problem with giving steroids in this situation is that it affects all components of the military. We just want certain cells to calm down. We want certain cells to calm down and stop producing all that noise. But we still need the immune system. And steroids are not a good choice for this. So there are some specific medications that work on this immune system component. And what they do is they either stop the chemicals that are being overproduced by these cells. These chemicals are called IL-6 or IL-1 beta. So IL-6 medication is called anakinra, and the IL-1 beta medication is the other way around. IL-6 medication is called tocilizumab, and the IL-1 beta medication is called anakinra. So you have these medications that have been tested. You also can use antibodies to try and help the immune system to fight. And we'll probably talk about that in detail about the convalescent serum. And one of the simple ways to do it is by using what's called as IVIG. You have immunoglobulins from people who donate their serum, and we isolate them, we purify them and give it. So IVIG is another option. So these are some of the options to try and address the immune component of this whole process.
So would you agree? I mean, generally what I've told people is there are really two main focuses. One is, how do you stop the virus? And then the second part, and probably the one that kills the most people, is how do you calm down the overactive immune system once it starts to really get going? And, for me, it's quite remarkable, unlike a lot of the other infections that we've seen, we don't see this degree in the flu of our own immune system then going haywire almost. And, as you alluded to, these molecules, IL-6, IL-1, the cytokines, like you and I will use in our terms. But, so really two parts of, I think, one is, how do you prevent the virus from doing what it does? And early on you mentioned chloroquine, and I think it's important for people to know the azithromycin, I think, is more there to prevent secondary infections when you're ill. But when it's after a certain period of time, when you're going on the ventilator, when you're requiring even heart-lung machine support, then really, you're looking at the body's immune system being overactive, and you may not have that many viral particles. So, that's how I've always split it up. Now, you've mentioned remdesivir. That's a very popular one that got a lot of press with Gilead. But let me ask you, which is very similar. They're all analogs of RNA. And so remdesivir is an analog of guanine, which is, again, a type of RNA, one of the 4-letter codes that go into RNA. And ribavirin's, an adenosine analog, I believe. Why would you use remdesivir over, ribavirin, for example? Because ribavirin, we have quite a bit of. It's FDA approved. It's out there. From your perspective, and this may be a little bit more specific question, but we do have a medicine similar to remdesivir.
No, I think it's a good question. So ribavirin does work on hepatitis, and some of the hepatitis virus, particularly the hepatitis C virus, can be treated this way. The problem with the ribavirin is that it is not very specific for the viral activity. Yes, you're right. They all are what we call as nucleoside analogs. They go and interfere as the virus is copying itself. It goes and sits there and gives a wrong. Exactly. Yeah. So ribavirin syncytial virus are called as RSV and hepatitis. But there were previous studies done with MERS, with the Middle Eastern Respiratory Syndrome virus. There was some response, but it is not as overwhelming. And so when this came along, when ribavirin was being developed, it's more historical than scientific, so to say. And when the ribavirin was being developed, remdesivir wasn't around. So now when the COVID-2 came along, the ribavirin wasn't a good candidate because all the studies, the New England Journal of Medicine publications, which show that the ribavirin may or may not add a whole lot to this. And that's the reason. And it has to do more with the specificity of where it binds. And it may be good for other viruses, but not a good fit for this particular coronavirus.
Now, we have a few questions from, I think, they're nice. Somebody asked, does the viral load actually determine how sick you are? So, in other words, I think what they're really asking is, is this why doctors and nurses are falling sick at a higher rate? Because they're getting a larger dose of the virus, or is it just hapstance? So what is your explanation for nurses and doctors being, again, the higher rates of being much sicker than the general population?
I think the one single word I would say is PPE. It's the PPE or lack thereof, right? So I think clearly, yes, I think there is hypothesis, and very strong hypothesis. It's not been proven. We've not been able to quantify this virus in this particular setting. But and if you go back again to the previous SARS and MERS epidemic, the mortality rate for healthcare workers is also high. It was in the 15 to 20% range. And studies from there suggest that it's very likely because the doctors go in, they get an immunity, and then they get a load of the second one. That this virus is slightly different from the first one, and the immune system is continuously getting overwhelmed. And that's part of the reason. And so if you think about it, the load of the virus that you're exposed to is high. And so you would think the viral load is higher as well. I think it's point on question, and that is something that all healthcare workers should be aware of. And I think the best way to prevent this is using proper PPEs, whether it's gloves, mask, face shields, goggles, which have tight fitting, because this is a virus that enters through the nares, the mouth, and the eyes, and your hands as well, because you touch things. So I think proper protection is very important. And there is another area where you see that this may be at high risk in ER, emergency rooms, because you don't know when they're walking in the coughing ICUs, when they're trying to intubate patients. And And because a lot of things that come out of the mouth and nose, we call them aerosols, right? So they are small things that you don't see. And wherever there's a high chance of aerosolization, the virus can get into you. If you do this repeatedly day in and day out, you're exposed to a much higher load.
So you think yes, you have repeated exposures, but even if you match for that, it seems like the healthcare workers are getting much sicker, which is interesting. Let me ask you, maybe I'll segment it this way. So if you're looking at drug therapy and you had to bet today, if you could pick 2 drugs and you had your wish, of all the drugs that are out there in development, which one would you say has the greatest potential?
Well, if you ask me
the most sense, what makes the most sense?
What makes the most sense? I think the antivirals make the most sense because you want to prevent the population. But there are 2 components to it. You also want to make sure that by the time these antivirals work, you have something to protect. And one of the best ways to do that is to making sure that they have immunity, whether it's active immunity or passive immunity. And active immunity, you have to wait for those 10 to 14 days, right? For the immune system to go through the process. So passive immunity, I think, has a great potential because it's immediate. You don't need to go through an FDA approval. And as I told you, 80% of the people recover from this. Everybody has to go through it. 80% recover from this, but I think the people who recover have a great service to the others, to help the others. Once they recover, once they have antibodies against the virus, which they have recovered because they have antibodies against the virus. I think has the immediate potential for trying to save those 15% who might sick, and you want to prevent them from getting sicker and going to the ICU. So to me, it's all immediate. It would be antivirals and the convalescent serum.
So while we were talking about convalescent serum, do you want to give our viewers an overview of the convalescent serum thought process? I know there are some questions about that, and as well as vaccine. So if you wouldn't mind, maybe in lay term explaining, how does convalescent serum work? How does it help? What's really required to make it successful?
So it's a very simple process. I mean, blood has several components, right? So blood has cells, the white cells, red cells, and platelets. These are the cellular component. Then it has a lot of proteins. The major protein is a function of how you eat and how the liver does. It's albumin. The second fraction of the protein is called globulins. And a big portion of the globulin is your immune arsenal. It's all those thousands of bugs, or I would say billions of bugs that your body is exposed to, and you have antibodies against those. And in some ways, it's like a one arrow for one bug, right? So, and this immunoglobulin portion, or called the gamma globulin portion, is very important. It's actively, you're increasing the armamentarium every time you come across. So what happens when people get infected with this and they develop antibodies, and it takes about 10 to 14 days for this to happen, you have the right arrow that is produced for this virus, and it goes and becomes part of that armamentarium of gamma globulin. So when you take blood out of people, you're able to separate out the cells and separate out the proteins, and that portion is called the serum, right? That portion is called the serum with the proteins, and you're able to take that and give it to another person. The big thing here is that you don't have to really worry about a lot of matching. You need to know what your blood group is so that you don't have the corresponding antibodies, because antibodies are part of that gamma globulin. So you want to make sure that if you have blood group B, then you have an anti-A, right? So you have to make sure that those things match. And you take that portion of the component of the blood called the serum from people who have recovered from this viral infection and donate it to the others. So they get immediate antibodies that is produced by the other people who have recovered, and that can help them in fight against the infection.
And that, again, this is one of the therapies that we're seeing more and more on the clinical side is the serum. Now, I think, one of the things that's important to know is that the serum has a particular level of antibodies, and ideally you want to get a high level of those antibodies before you give it to somebody, and you also want to make sure they work. And there's a lot of testing that goes around it. I think there's a couple of questions regarding this. One is, should the antibodies be given before a person gets seriously sick?
Absolutely. Again, a point on. Yes, I think ideally you would want to, in this whole series of things, you want to make sure that the body's immune system is supported before it goes down the slippery slope. I really think that convalescent serum's role in preventing people from getting sicker is very early on. Yes, we are desperately trying everything in people who are in the ICU. By the way, the ICU care is in the hands of the outstanding ICU doctors, without whom we wouldn't know what to do with how to handle the lungs that's struggling, right? So there is definitely a role for convalescent serum, but before that stage, I think clearly that's where the convalescent serum really has a role in trying to make sure people don't get sicker.
Okay, and another question you have is, we have, and I'm not sure how to answer this. Why can a person who has antibodies but not got a positive test not be able to donate plasma? Is the virus mutating?
Can you repeat that? I didn't get it.
Yeah, so to me, it's a little confusing. Why can a person who has antibodies but not got a positive test be able to donate plasma? Is the virus mutating?
Not necessarily. So there are many reasons. You may have to repeat the test once or twice. It depends on where the testing is done. At least most testing so when you test for the virus, you test for a small portion of the RNA, and you use a technique called RT-PCR, right? So when you do the RT-PCR, you know that portion. Sometimes the sequence changes, and because the sequence change, RT-PCR you might think doesn't work. So, however, it's not just because of the sequence. The coronavirus mutates, but not as much as, say, flu virus, for that matter. Number two is it has a sensitivity of testing. Depending on where you take this sample from. For example, the nose area is one area. That technique is also very important. In fact, our nurses learned to say when they put a swab inside the nose, and that's the most sensitive area, they have to make sure that it gets as close to the ear. That's the technical detail to it. If you don't do it, you don't get the right amount of particle. So in other words, you have to do the technique correctly. Now we're also trying to look at testing in the mouth, in saliva. People from Hong Kong and Singapore have shown that you can actually take it from the throat, which is much easier. And you can also get it from fecal samples, from stool specimens too. So I think the testing needs to be looked at a little bit more carefully. That's one possibility, right? The second is that by the time, and it's a 14-day period to produce the antibodies, and it's about a 28-day period when their antibodies are going up and the virus levels are going down. So depending upon where you are in that process, and by day 28, sometimes you may not see the virus. You'll only see the antibody, right? So it all depends on when you are actually infected, not when you were tested, but you actually got infected, which might be sometimes even earlier than the day you got tested. So there are these technical details to consider.
There's another question, and I think you've seen it also. So the moment a patient gets into the ICU, would you recommend the patient getting convalescent serum? So again, one of the things that's difficult, and I'll be a little technical here, is I've done a lot of reading on this. And I have good friends who are immunologists like Dr. Swamiji. When you collect convalescent serum, it's very important to collect enough antibodies. In the United States, the current way that the protocol is written, we are actually not testing the amount of antibodies that are present in the convalescent serum. We actually don't know. The study out of China, which looked at convalescent serum, actually measured what we call the antibody titers. In other words, how much is it there? And the usual level is 1 to 160, and we don't test for it. Actually, by the way, the current protocol in the United States is that's run through the Mayo Clinic. Number two is we don't do what's called a neutralization assay. In other words, we don't actually test whether this antibody that's in this convalescent serum is going to work or not. And that's just, we may or may not have a good or bad assay, but that's also not there as a part of this protocol. So I want to be a little bit cautious when we're recommending convalescent serum to people and everybody out there, that this looks like a panacea, but may not be. It can still not work in certain people. It can cause, in rare cases, the opposite effect and hypersensitivity reaction, which means an allergic type reaction to the serum. So, we're a little cautious on saying, who would get the convalescent serum. Right now, I can tell you the people who have been sick on the ventilators, typically within Houston and, from what I've talked to with my colleagues around the country, are the ones who are getting convalescent serum. Just because, again, the process is not perfect at this point. Comments, Dr. Swamiji?
No, I totally agree with you, Dr. Pranavji. I think you don't know the product, right? So you're assuming that that's good. Since you recovered from the virus, you may have the antibodies. In fact, the study that just came up on preprint in Lancet, they actually measured antibodies in about 195 patients. Interestingly, they did a lot of correlations, and they found that not all of them have the high-level antibody. And they did neutralizing antibody testing and various antibodies against the spike protein, the envelope protein, etc. And they've actually found that there are about maybe about 15 to 20% of those patients, actually their titers are very low, but yet they recovered. In other words, there are other things that we don't know of other than the antibodies, right? So there's also that to be considered. So for now, I think convalescent serum is very reasonable. And you mentioned the vaccine earlier on. Until we get to that, and we are probably about a year away from it, at least. I think this might be a way to do it. Eventually, I think this is going to be fine-tuned. We'll know more about what the quantification might be, how much antibodies there are in the serum. And we might be able to pool some of the sera from different people and maybe enrich the sera. There is also a company called Takeda, which is trying to isolate these antibodies and purify them and give it, like I mentioned earlier, with a product called IVIG. So this would be COVID-specific IG that can also be given. So it's going to be a lot of fine-tuning of this process.
All right, so I'm going to take a little break and try to see if you and I can answer some of the questions that have been put out there. We have a few questions. So, and I'm going to take them in order. So is it true that lupus patients are not getting infected by COVID-19? So it's a very interesting question because as people know, a lot of lupus patients are actually on hydroxychloroquine and they do have some immune suppression. And I'll be honest, I don't know the data as far as lupus patients, Dr. Iyer.
So there are different theories, and there's my own hypothesis in which you're writing a manuscript on, and I can tell you more if it is at least pre-published about it. But interestingly, it has to go back to the idea that the innate immune system probably plays a role, and the way these viruses are kind of gave you the analogy of the immigration officer, and there are various kinds of immigration officer. The immigration officer for this particular RNA virus is a toll. It's like a toll booth, right? It's toll-like receptor number 7. And interestingly, toll-like receptor number 7 overactivation is what causes lupus in women. And it's typically seen in women because there are two X chromosomes, and both X chromosome and these X chromosome related genes. And many of the patients with lupus are on hydroxychloroquine. So there may be something to it, and we don't have any data to it. But when I went and looked with a fine comb at the type of patients in the Chinese study, the Italian study, there were a handful of people with rheumatological conditions. A lot of patients with diabetes, with other kinds of comorbid conditions, if you will, including cancer, but the rheumatologic conditions are few and far in between. So there could be something to it. We need to study this in detail.
Another question, which I thought was really nice, is how can we make a suitable vaccine if the virus is constantly changing?
So the virus is not constantly changing. And I think when we talk about mutation, it's trying to survive itself. within the constraints of the environment that it is in. For example, the virus that from Wuhan is not the same virus that's traveling through the United States because it had to adapt itself. But once it's in a community, it doesn't change a whole lot because it's easy. Once it finds a living being to latch on to, the change doesn't happen too much. There's not too many evolutionary pressures to do that. That being said, so there are many ways to approach the vaccine. And the first vaccine that everybody saw was a lady on TV who got a shot on the arm. Was the exact copy of this virus mRNA, and the idea is that these pieces are going to be taken up by this innate cells and probably mount an immunity. I really think that there are several aspects to this, and if you go back to the SARS era, there are vaccine elements that can be derived against proteins on the outside that is covering the RNA piece, the spike proteins, and where this virus latches onto the cell and enters the cell. That's called the receptor binding domain, where it tries to enter the cell. So that's another area potentially you can identify that region and develop some kind of a protein against it or antibody against it, and of course mRNA itself. So there are all kinds of thought processes right now. I think it is going to happen, and it's one of the other things, and I think based on the SARS theory, and we still don't know that yet, is once you have a vaccine, it's very likely going to last for a long time. There was a 6-year-long study from the old SARS epidemic. They looked at antibodies that lasted for as long as 4 to 5 years, and the antibodies went away, but then you still had memory cells that seemed to be active. Of course, we never saw another SARS again. So it's quite possible that at least from this 6-year follow-up, that these vaccine effects can last a long time.
So another question from one of our viewers. I'm a pediatrician and had a question. Why do patients respond differently? Some younger patients less than 50 years old with no comorbidity are getting significantly worse. So why is it that the young, I'll add a little to that question. Yeah. Why are the young relatively spared and the elderly not? And then why are there exceptions to that rule?
Right, and it's very different from the influenza. Influenza affects the very young and the very old. In corona, one would think that it's a very similar feature, but the younger, the kids, you don't see a whole lot of problems. They do get infected, but the effects are not so bad. And the older folks, of course, are very vulnerable for various reasons. The Queen Mother, you all saw, is 94, 95. She's also positive, but you saw her speech. So not every night. And I think there are other reports of a 100-year-old recovering from this. Interestingly, they're all women who recovered from this. So there may be something to it also. But why, going back to the point, the younger people, why do they get affected? I think it has to do with the immune signature. I think it has to do with how the immune system tries to overreact. And that immune signature, that's where I think the key is. Why would a virus that goes through it tries to have to go through the recognition pathway, but doesn't do so, and the immune system tries to overcompensate? I think it has something to do with that. And we see that in our own setting, in the setting of cancer. I treat certain types of lymphomas called T-cell lymphomas. And we see a situation where these cells just take off for no reason, and they produce a lot of cytokines. We call it HLH, or hemophagocytic lymphohistiocytosis, for HLH. So in some ways, this is very similar to the HLH that we see. And this cytokine storm, I think, is because of this macrophage subset activation. You have other cells that are trying to overcompensate for this. So somehow, somewhere in this younger population, there is overactivation, and that needs to be looked at carefully.
Now, some, I think, more basic questions. When should a person be tested? When he or she gets a fever? Or when are we recommending testing? I think certainly if you're getting a high fever, you've got a cough, definitely if you're getting shortness of breath, it's a good idea to get tested. It's not just to know that you have the virus or confirm that you have the virus. Also, it helps you know whether you are potentially infectious to somebody else with this. I mean, I tell my patients, in this month, this time, if you get a high fever, the chance that you have COVID-19 is very, very high. There are very few other things that will do this to you statistically. So certainly you should get tested, if you're getting high fevers. Another question is, can the virus pass on from the dead body of an infected person? Dr. Iyer.
So as long as, the virus is not going to live for more than there's no living being. But having said that, the aerosols can live, and the aerosols, there's a very elegant study that is done where they put these aerosols in different, in copper and cardboard and plastic and steel. And I think in steel, it actually lasts for 3 days, and cardboard for about 24 hours. So if you think about it, the dead body, it's probably going to be somewhere in between. And I think I know why this question is coming from. And unfortunately, life happens, and a lot of people and their families, things happen. And how do you handle those situations when there is a death in the family, for example? But I think the advantage or so is there are ways to prevent that from happening. But to answer your direct question, I think it's probably going to be in the span of within a day to 3 days. The same thing that you would see in any inanimate object if you don't do anything about it, in other words. But on the other hand, if you try and sterilize it, use formalin fixation, so those things, then it won't become an issue.
Now, another question that people have, and I think this is a general question, but it's a good one. What are all the symptoms? Should someone be worried if he or she has a dry cough for a few days? So again, I think if you have a dry cough, you have fever, you have shortness of breath. Now, we've also recognized that a lot of people will not have those, but they may have gastrointestinal symptoms, diarrhea in particular. So, those are all potential symptoms of COVID-19. Again, where should you really be concerned that, you're thinking of going to the emergency room is really when you're getting short of breath. If you're getting shortness of breath, when you're moving around, if you're lucky enough to have an oxygen meter at home and you're getting short of breath, if your oxygen levels are going down, you absolutely need to go to the hospital. But you can certainly have all of those symptoms.
If I may add to that, I think one other area is, of course, travel, right? So I think travel has come down quite a bit. It's been curtailed. But if you're exposed to people who have been traveling, and you have a lot of people who go to work, not necessarily to the hospitals, but grocery stores and whatnot, and you interact with a lot of people, I think that's the unknown too. So if you do have these symptoms, potentially you should talk to your physicians, and if you can, on the phone, right? These days it's all telemedicine for the most part.
Do you think we are through the worst of the pandemic in the United States?
I don't think so. I think we still have to see we're not seeing the inflection point. We're not seeing the peak of it. And I think I'll show you one other slide here, if possible. And the inflection point is based on a lot of things. It's based on the doubling time. And if you look at the doubling time here in this picture, where is it here? So this is the doubling time. This is the coronavirus. There's this engineer called Mr. Robert Williams who does this on a daily basis, and great work that he does. This is coronavirus cases per capita over time by country, and this is on 8th night. And you can see the US graph, that's the blue graph, that's going up here. And you can also see the writing here, it says the doubling time is about 8.4 days. And the per capita growth is about 9%. You can start seeing that it's slowly trying to bend itself. It's not happened yet. So which means, and these two lines in the top, that's the capacity. The top capacity is when the health system is going to be overwhelmed. And that's where I think is going to be a problem. So 8 days from 8th, so about 15th and 16th of April, 17th of April, that's probably going to be in the peak that everybody is anticipating. It could be a little, few days, a week or two later in Texas, where we are, but it could be around the time in say in the Northeast and also in the Northwest.
No, I think that, to add to that, one of the important things is also to note that it's also very regional. So New York City may be one area where, there's a huge pandemic going on, capacity peak. It may be very different in Nebraska. It may be very different in Houston, Texas. Certainly in Houston, where we are currently, our peak is estimated to be April 22nd. There are other places where the peak is estimated to be in May. And then there are other places which we won't have a good predictive model if we suddenly have a blossoming of cases in a particular area. So again, I don't think the pandemic's over. I mean, I honestly think that we'll still be worried well into late summer, into July, August. I think Dr. Fauci said something very interesting. He said this is probably the end of the handshake in the United States, or the death of the handshake. And he may be right. This will continue to be a problem, I think, until we have a vaccine or we have a large enough portion of the population that's immune, and then you get this thing called herd immunity, where the virus doesn't spread as rapidly because you have a lot of immune people who don't keep passing the disease on. So I think that's here to stay. There was one follow-up question at what is the ideal oximeter reading? Anything above 93% should be okay. Any preventive therapies for COVID-19, Dr. Iyer?
So I think the best prevention is the social distancing, hand washing, personal protection. Once you do have, so that's primary prophylaxis, right? So and secondary prophylaxis is after you encounter and have positive for the virus, and there are ongoing studies. That's where, as I mentioned earlier, hydroxychloroquine may play a role. There's a very large study in Minnesota that's on its way, and I think they've recruited about 500 of their 1,500 patients. So, and that's hydroxychloroquine versus placebo, and these are actually people who are not yet fallen sick, and some of them are not even turned positive. So this could be a right opportunity to see if this works in that particular setting. So I think other than social distancing, hand washing, etc., and everything that you do to make sure you don't come across it, hydroxychloroquine may have a big role.
Okay, and then I think one last question. Is it safe to have delivery and eat food from restaurant after reheating it?
I think so. I think it's food, and as long as you heat the food, I think it's reasonable. What becomes a problem is the container and everything. I think now every one of us has developed all kinds of rituals when you go to the grocery or get food from outside, and you wipe it clean, and you have the soap solution and whatnot. So I would do it until we know that we are safe from the coronavirus for the most part.
Dr. Iyer, anything else that you would want the audience to know about therapies and coronavirus? I think, summarizing, we can say that, there's prevention, which we've talked about, and unfortunately there is no medicine. So we're back to our traditional ways of preventing infection, especially social distancing. Once you have it, there is no known therapy. There are therapies that are antivirals that are on research protocols. Remdesivir we mentioned today. We're also talking about hydroxychloroquine, and you eloquently explained the mechanism, how it works. And then if you get really sick, perhaps plasma therapy, with convalescent serum. So anything else, Dr. Iyer, that you would want the audience or any of us to know as far as the therapies in COVID-19?
No, I think you've summarized it very well. I just want to add one point. There are many other things that are not investigated. For example, the role of the heat and temperature and ultraviolet rays. There are all these studies in the near ultraviolet, or the far ultraviolet, I should say, not near, in the 200 nanometer range, which are actually not harmful for the humans and have shown to get rid of influenza. So these are something that can be deployed in a large scale setting. So there are those things that also need to be looked at instead of trying to find a soap solution and wash your hands. It might be something for a large scale operation. But I think the most important thing for the people out there is be aware. Don't be taken in by those things that circulate, and particularly in the social media and WhatsApp and other areas. Yes, those are interesting, and I know a lot of people are at home and some form of entertainment, but ask questions. Do not just blindly go and accept something. And this might be the right forum, but I think there's going to be a lot of such webinars where we'll be able to ask this question. And the science is rapidly changing. So what might have been said a week ago might be different 2 weeks from now. So keep your eyes and ears open, and stay open. And at the same time, I think do your bit if you possibly can. I think the idea of convalescent serum now is probably one of the best things you can do. Somebody who's recovered from this, try and contact your hospitals around you to see if you can be the right donor. And all these hotlines that Kavita ji can provide. And second is that support. And I think the community support in trying to do such things, whether it is making masks, whether it is supporting a cause, coming up with fundraisers for a project. I think this is the right time to do it.
Thank you. Kavita ji, I see you online, and any comments? Or?
Thank you, Dr. Swami, and thank you, Pranav. Great discussion again. It's just that I learned so much myself, and this gets shared so many times over. It really is a very resourceful tool for everyone that we know and that follows Sewa, and I create a community. And thanks to the inspiration of all the doctors who've been involved, Sewa is now stepping up in towards the direction where we can actually make concentrated efforts, whether it's seeking donors or finding people who are possible donors, maybe even connecting them with families that need plasma donations for their loved ones. So I think it's a great way that the community has come together through knowledge. And thank you all for sharing that. We are just wiser because of all the help we get through all the doctors and other great supporting mentors we have at Sewa. So thank you. And we look forward to talking to both of you again. Maybe, as you said, it's changing every few weeks. the whole thing just changes. The concept changes, the approach changes. So hopefully next time we'll have something more to add to our conversation. We keep getting questions, Dr. Swami, Dr. Loyalka. So we'll again keep those ready for you next time. My daughter wanted to ask, can we order pizza and eat? She was like, probably, can you please ask that? I was like, okay, let me ask that question. So
she can definitely order pizza and eat it, just reheat it once it gets there.
Right.
Okay. All right. So we can do that. Good. I'm off kitchen duty then. Okay, Shri, thank you so much. Can you take it from here?